All right, in this video, we're going to focus on hormones in general too, and just kind of talk about what hormones are, what can they do, and then we're going to really get into how do different types of hormones function. So we're going to look at really two main types, which are the lipid-soluble ones and the water-soluble ones. So you may see them called steroid hormones, and peptide or protein-based hormones is usually like the other way to make that distinguishing factor between the two. So we're going to talk about kind of how they're different in terms of how they affect the cells that they target, right? What do they do? And then get into, how do we help regulate that, right? How do we help regulate hormones so that they're not just stuck in the body all the time? So first and foremost, before we get into all of that, we really have to define what a hormone is, right? What is a hormone? And I know I say that and you're probably like, I think I know what a hormone is. But if you had to put it into words, right, what is it? So really it comes down to this. I'm going to read what we have here in bold, and then I'll kind of give us a quick and dirty definition as well. So hormones are chemical messengers that instruct various tissues on how to behave in order to affect a specific physiological outcome, such as metabolic change, growth, reproduction, etc., to maintain homeostasis. Basically, what it means is it's a chemical that's going to tell a tissue what to do so that we can maintain homeostasis. That's all it is. It's a chemical that tells other cells, other tissues, other organs what to do to maintain homeostasis. Quick, easy, not too crazy. And so when we're using a hormone to tell a cell or tell an organ or a tissue what to do, that could be we're telling the tissue to make new molecules. It could change the permeability of the cell membrane so that the cell absorbs something or releases something. It could affect the metabolic rate of certain things. This can be used for the contraction of certain types of muscle, like cardiac and smooth muscle, right? And this is just a short list of actions. But basically, chemical that tells the cells, tells the tissue, tells the organ, do X, right? So that we can maintain Y. And we can classify them again based on whether or not they're soluble in water. Lipid-soluble hormones, sometimes you see these called steroid hormones. That's usually the one that people think of. But the big thing is they're lipid-soluble, meaning they are kind of like non-polar, right? They're not going to dissolve in the water very easily. So they're going to need something to help move them around so that they don't glob together. So types of lipid-soluble hormones include steroid hormones, which is probably the one you've heard of in a prerequisite class. So things that have a sterone, right? Steroid in the name. So things like aldosterone, cortisol, cortisone, testosterone, estrogen, right? Those are all steroid-based hormones. They all have the same. They all basically use cholesterol as its backbone, and then we have little functional groups that come off of that backbone. It also includes the hormones found that are produced by the thyroid, right, and whatnot. And then with the water-soluble hormones, these ones are free. They don't need anything to help them move around because they already can dissolve in water. So we don't have a concern that water-soluble hormones will glob together because they'll freely dissolve in the watery plasma in the bloodstream. And so things like amines that come from amino acids, or the one that you probably are more focused on or have heard of before, which are the protein-based hormones or peptide hormones. The eicosanoids, which are just prostaglandins and whatnot, I'm not going to be worrying about those so much. So we can really kind of put it down to the steroid-based hormones and thyroid hormones and the protein-based hormones. Now, if you are a lipid-soluble hormone, you're nonpolar, because you're nonpolar and you're lipid soluble, you actually can get into the cell very easily because the cell membrane is lipid based, right? So if you are a lipid soluble hormone, you come out of the bloodstream, right? You escape from your transport protein, you escape the bloodstream, and then you go directly into the cell that is your target. This means that lipid soluble hormones are able to directly influence gene regulation and gene expression. So if we kind of sum up, because I know looking at this and reading this, you're like, what is going on? We have a lipid-based hormone. It's moving through the bloodstream. The hormone leaves the bloodstream. It passes right through the membrane into the cell. It joins up with its receptor. So the hormone and the receptor bind inside the cell. And then together, the two of them, the hormone and receptor bound together with what we call a hormone receptor complex, are going to actually get into the nucleus. They're going to bind to the DNA and directly influence whether or not that gene gets expressed. So we can like induce something, right? Hey, we're going to turn this gene on, we're going to express this gene, or we can repress it, we're going to turn it off. So we directly impact which genes are getting used and how often, which then of course affects what the cell does, right? If we turn a gene on to produce a certain protein, that cell is now going to make that protein more than it was before. Or if we repress it, right, that protein that gene would normally make is now going to be in a smaller amount, right, if it's made at all. So the takeaway here is if you look at the picture, right, I know the text here is maybe a little bit complicated. If you look at this picture and you can tell me those bam, 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 those 4 steps, I would be very happy. So again, this is for lipid soluble ones only because remember, the plasma membrane is lipid based. Like attracts like in this instant, right? In that the lipid soluble hormone that is nonpolar and hydrophobic is going to have a very easy time getting through a membrane that is also hydrophobic, that is nonpolar. So here's some examples, right? The steroid hormones, again, you see that 4 fused carbon rings, that's our cholesterol base. And so this is going to include things like aldosterone, cortisol, cortisone, estrogens, testosterone, progesterone, calcitriol. And you'll notice the names, not all of them necessarily, but a lot of them have some sort of sterol, sol, sterone. Stro is like in the name, right, for most of them. And so if you see that, you can kind of already have an idea, right, of what they, if they're lipid soluble. Whereas on the thyroid side, we're going to look at like T3 and T4, triiodothyronine and thyroxine. I'm perfectly fine with you just saying T3 and T4 because I know spelling those is not very fun. But you should be able to recognize the name in its entirety if I use it. Now the water-soluble hormones, the polar ones, right? Because they're polar, they cannot get into the cell directly. Big difference here. So that's the main thing between the water-soluble and the lipid-soluble. Lipid-soluble can get through the membrane, but the water-solubles cannot. And so when they bind with their receptor, that has to be on the membrane. So the polar hormones, the water-soluble hormones, they never actually go inside the cell. That's a big difference between the two. And you should be able to tell me that, right? You should be able to tell me that for sure if you're comparing these two, that water-soluble and lipid-soluble hormones have this difference in terms of where they meet up with their receptor. Lipid-soluble meets the receptor inside the cell. Water-soluble meets the receptor outside the cell. Now we bind with our receptor on the outside of the cell, but that's not the only, it's not going to do nothing, right? That's going to cause a change inside the cell because the receptor it spans the whole membrane. So there's a side of the receptor that is inside the cell, and we're going to go through and basically go through a chain, kind of turning on these secondary messengers, right? And that's what's going to cause the effect. So we go through our little figure here, right? Our water-soluble hormone leaves the bloodstream. It binds with the receptor that we see on that membrane. This is going to then join up with a, kind of activate a G protein with adenylyl cyclase, right? ATP gets utilized in this process, which is then going to activate CAMP, right? CAMP, right, is then going to serve as another messenger to activate protein kinases. Remember, kinases are going to be enzymes that add a phosphate group that usually turns things on, right? So it's going to phosphorylate or add a phosphate group to something that usually turns something on. So we have like this little chain going on, like this chain of events happening inside the cell. So what are some examples of water-soluble hormones? Epinephrine, acetylcholine, insulin, right? These are all protein-based. Like insulin is 2 peptide chains that come together. Because it's protein-based, it's not going to be able to pass through the membrane. It's not lipid-based. So here's just some examples. So like our amines, we have like epinephrine, melatonin, histamine. Those are going to be ones that are amino-based, right? We have an amine group on them. And then the peptides, the proteins, right? Those are going to be everything that comes from the hypothalamus, right? This is going to be growth hormone, TSH, ACTH, FSH, LH, prolactin, MSH, insulin, glucagon, parathyroid hormone, calcitonin, gastrin, EPO, leptin, prostaglandins, right? It's a super, super long list of hormones, right, that are water soluble. So really, I mean, if you had to guess, most hormones are going to be peptide based. We had a very short list on the lipid based. So our water-based hormone is going to bind with a receptor that's embedded in the membrane, but what the heck is going to happen on the inside? Let's take a walk through what these water-soluble hormones do. What do they trigger inside these cells? So a lot of times we have the receptor, right, that's going to bind with our hormone, and hanging out, right, with that receptor is what we call a G protein, right, or a G protein coupled receptor protein. So basically this is a receptor that has a G protein. The G protein has 3 parts, an alpha, beta, and a gamma. And that when the hormone binds to the receptor, we're going to basically activate that G protein, right? And we see that there. We go from GDP to GTP. We've activated, right, when something binds. That is going to allow us to form CAMP, right? CAMP is going to be a second messenger. So basically it's like going to carry that signal on, like, hey, we're turned on. That means there's something bound to that. It gets activated, so the G protein basically helps form CAMP because we've activated the enzyme adenylyl cyclase. So if we're walking through what's happening here, because I know like this sounds like a lot. Hormone binds to the receptor. When the hormone binds to the receptor, our G protein is activated. When our G protein is activated, it will activate adenylyl cyclase to produce CAMP. That's where we're at right now. CAMP then will tell other proteins what to do. Namely, it'll phosphorylate, right? It's going to add a phosphate using kinases. Remember, kinases, that's an entire group of enzymes that their job is to add a phosphate to turn things on. That's what I would like you to be able to tell me. Receptor or hormone binds to receptor. This activates the G protein. The G protein activates adenylyl cyclase, which then forms CAMP. CAMP then works with kinases to phosphorylate other proteins to turn things on. I think when you think of it the way I just said it, it simplifies it down in a way that makes a lot more sense and feels less overwhelming. So we phosphorylate. Again, remember, when we add that phosphate, right? This does use ATP to do this. That's where the phosphate's coming from, right? And usually, again, adding a phosphate or removing that phosphate is going to cause the protein that you're adding it to or removing it from to change in shape. And so again, usually adding a phosphate using a kinase turns the protein on. It activates it. When you remove the phosphate, dephosphorylation, this is done by a phosphatase, and it usually turns something off. That's what I would like you to remember from what we have here on this slide. That's what I'd like you to remember. Remember, phosphate typically acts like a switch for proteins. You can turn it on, it can turn it off. Adding the phosphate usually turns it on, and we can do that using a kinase. Taking the phosphate off usually turns things off and is completed by a phosphatase. There are other things, though, that other secondary messengers besides CAMP that we can use, including IP3, DAG, and calcium. So, our hormone binds to our receptor, right? We have our G protein that can activate a phospholipase C or PLC. Hormone binds to receptor. This activates our G protein. The G protein then activates PLC, and in this case, instead of making CAMP, because it's not adenylyl cyclase anymore, we're activating PLC instead, the downstream products are different too. So PLC then will produce the IP3 and DAG. IP3 will cause a release of calcium from the smooth ER. Calcium will activate calmodulin. This then helps activate kinases to turn things on. So again, it's very similar process to what we talked about earlier. It's just we've changed the main players here. You should be able to kind of tell me a little bit about both. In the very least, like, they have a similar thing in that we have the hormone binds to receptors. This turns on the G protein. G protein activates something else, which then activates and releases secondary messengers. Bam, bam, bam. Right? Whether we're looking here or we're looking at the other one with the adenylyl cyclase, it's the same kind of concept here, right? It's the same concept. We also have what are called RTKs, or receptor tyrosine kinases. So, what does this mean? And I know, like, we're getting a little crazy, right? And you're like, bring back the lipid soluble ones. Those ones were easier. It's okay. You'll make it through. So, in this one, you have these two receptors, right? That are kind of like they form like the halves, right? So you have one receptor, but it's split in half, right? Each half being an RTK. When the insulin, in this case insulin, I'm just going to use that since it's our example. When the insulin comes in and starts and interacts with the two halves come together to form a whole. We call that a dimer, right? Because it's the two pieces have come together. So the RTKs come together, right, to form the whole receptor. And then they're going to autophosphorylate. That means they're going to phosphorylate themselves. They're going to add a phosphate to themselves. So insulin binds, the RTKs come together, they phosphorylate themselves. This then turns them on. Remember, what does phosphorylation do usually? It usually activates something. So from there, they can then phosphorylate themselves, but they also phosphorylate and turn on a signaling molecule. And then that'll cause a cascade. In this case with like insulin, right, we can take like those GLUT4 receptors, we can tell the cell to, put those GLUT4 receptors on the membrane so that we can bring in glucose, right? But if you can tell me kind of like what's happening here with the insulin example, like in this picture, I would generally be pretty happy. Now, how far does the signal travel? This is what we call an action radius, right? Basically, how far does that hormone, how far does that signal go out? It can be local or it can be distant. Depends. If it's local, this can be autocrine, meaning we're going to self kind of regulate, right? We're going to tell the cell that released it to do something. So we look down at the bottom here on the left, we see the autocrine where the cell has released that hormone and then it just binds with it itself. So it's auto, right? You think self, like autobiography. So the cell is signaling to its own self. Paracrine is like just to the neighbors. So the cells that are in the local area. This would be like if you were to put flyers out in your neighborhood, right? You're going to stay within the streets that are around your house. Endocrine, what we know of as endocrine, is going into the bloodstream and it's going all across, right? Way far away. Most things, and especially a lot of the ones we're going to talk about, are endocrine. They're going to go into the bloodstream and they're going to move from one part of the body to a totally different place. But you do want to at least keep in mind and know the definitions of autocrine and paracrine because those things do happen, right? We do have hormones that are more localized, right, that are going to impact the cells, either their own self or the cells around them. So what do the target cells do in response to that hormone? What kind of effects can we see, right? Especially because when you think about it, you don't just have one hormone being released at a time, right? You have a combination of hormones that are released all the time, right? You basically have a varying cocktail of hormones at any given moment. So what does a target cell do when it gets these combinations of hormones? Sometimes you have two or more hormones that work together. So we see that up here at the top. We see hormone J and K. We see that if you just have hormone J, you get a response. If you just have hormone K, you also get an OK response. But when you have J and K together, whoosh, much bigger response. So it's synergistic. The two hormones work together to produce a bigger effect than either hormone can kind of cause by themselves, right? Permissive effects. If you have hormone A by itself, you don't get much. If you have in order for hormone A to do much of anything, you need the permissive hormone. So, for example, progesterone. You can have a lot of progesterone, but unless you have the estrogen there prior, the progesterone's impact isn't very high. Basically, you need one hormone to kind of unlock, right, the cell's ability to be responsive to that second hormone. So the first hormone unlocks the cell, so to speak. The second hormone then comes in and does its job. And then antagonistic. This is where they do opposite things. So like insulin and glucagon work opposite of each other. Calcitonin and PTH work opposite of each other. Right, so we see our little graph here as hormone X drops over time, hormone Y increases. Or I should say, as the cell's response to hormone X drops, the response to hormone Y increases. So we see like they basically do opposite jobs, right? Is kind of what we think of there. We also can, change a target cell's response based on how many receptors the target cell has. So when you are a cell, you can only respond to the hormones that actually bind to their receptors, right? If you don't have the receptors, your cell's not going to respond, right, to any hormone in any amount because it doesn't have anything. It can't listen, right? You think of the hormone like the voice, the receptors are like the ears. If you don't have any ears, right, you can't hear the message in the hormone. You don't have any receptors, then you can't get the message out of the hormone. So you have to have the receptors for that hormone, and the volume of those receptors will also have impact. Now we can make new receptors, we can recycle them, and we can kind of change how many receptors are available to regulate, right, the cell's action, right, and how that hormone can respond. So receptors can be upregulated if we don't have a lot of the hormone. This will make the cells more sensitive, or we can downregulate. There's too much of that hormone, we just have fewer receptors. And so we can basically, the hormone receptor levels regulate the response because, again, the cell can only do something if the hormone and the receptor bind together. If you downregulate the receptor, you downregulate the response to the hormone, even if the hormone level is high. And then hormones do not last forever, right? Hormones have a lifespan, what we call a half-life, which basically means the time it takes for the concentration of that hormone in the plasma to be reduced by half. So if you had, let's say you have, I'm just going to make up an amount, it's not going to be realistic, but if you have like a gram, right, how long does it take to go from a gram to half a gram? How long does it take to go from half a gram to a quarter? From a quarter to an eighth, right, for that hormone to be reduced by half? This varies greatly depending on the hormone. So let's look at a couple examples. Something like thyroxine. Its half-life is 6 days. Versus something like insulin, half-life is 8 minutes. So you can see they really can vary, right? It really depends on the hormone. So we have a half-life for it to be reduced by half. We also have the liver, right, that can remove hormones from the bloodstream and kind of break them down into something that won't necessarily conduct the same function. We can also control how a cell responds on the hormone concentration. So let's look at this graph here. We see the relative hormone concentration. So the higher it is, the higher the concentration compared to the others. The lower the concentration, right? And basically, like, tissues will respond when the hormone concentrations are like normal. And if we go higher, like when we take them as a medication, that we can get different effects from normal, including side effects, right? But we see how they can change, right? That changes in the plasma. So here we're looking at over pregnancy. We see that HCG, right, that's going to go spike up around, kind of starts climbing around week 8, peaks at around week 12, and starts to really drop off around 16. Whereas estrogen and progesterone continue to kind of steadily climb throughout the pregnancy. Some hormones are secreted in short bursts when we need them. So we can see signals being sent from the nervous system, right? So this can be things like for signaling for epinephrine. When we get chemical changes in the blood, so the pH of the blood is changed, or the osmolarity, meaning, the water salt concentration, right, of the blood has changed. We can also do this using TSH or ACTH, which are going to help control the thyroid and the adrenals. And then doing this is going to help contribute to homeostasis. So like most of what we're talking about in terms of like the most of hormones, like what they contribute to in terms of homeostasis is doing it through negative feedback. It came back, right? We talked about negative and positive feedback a while back in the beginning of the semester. They're, like I said, they're not going away anytime soon. Most hormones use negative feedback, right? If we stray too far from normal, we're too high, we're too low, whatever, negative feedback regulations or negative feedback loops brings us back into the normal range. It helps basically do the opposite of the direction we were moving. Very few are going to go in a positive feedback direction, so I'm not as worried about that. You should be able to tell me some of the examples of the negative feedback loops, though. When we cannot keep these hormones in check, right? When we're not able to use these hormones to regulate things, either the hormone, a lot of times the hormone secretion has been disrupted in some way. Either it is too high or too low, then we get endocrine disorders.